Hi everybody, and welcome to our module on B cells. B cells are part of the adaptive immune system, and they are lymphocytes just like T cells and natural killer cells. And this is a picture of a lymphocyte in a blood smear on the screen here. There are millions of B cells in the human body, and each one recognizes a unique antigen. And once a B cell recognizes its unique antigen, it can synthesize antibodies, and these antibodies will attach to pathogens, and this leads to the elimination of the pathogens from the body. The B cell receptor is one of the most important parts of the B cell. It's found on the surface of the cell, and this is what binds to antigens of invading pathogens like bacteria and viruses and parasites. The B cell receptor is made up of 4 chains. There are 2 heavy chains and there are 2 light chains. And this outer portion here, which is made up of some of the light and heavy chain, is where the antigen binding site is. There's 2 antigen binding sites on a B cell receptor. And we sometimes refer to this outer portion as the FAB portion, that stands for fragment antigen binding. And we sometimes refer to this inner portion made up of 2 heavy chains as the FC portion. And if you think this structure of the B cell receptor looks familiar, you are correct. It looks like an antibody, and that's because B cell receptors are made up of IgM antibodies. This is an IgM antibody right here, and it's got 5 of these B cell receptors linked together. So the IgM antibody is a pentamer, but if you take a monomer, which is just one of these structures, that's the B cell receptor. So when the B cell gets activated, what it does is begin synthesizing copies of its receptor in the form of IgM antibodies. Here's a more detailed picture of the B cell receptor, and it has some key components that you should be aware of. So both the heavy and light chains end in these purple regions called variable regions, and they're called variable regions for obvious reasons because they vary from one B cell receptor to another, and this is the way we have so much diversity in our immune system and we can respond to such a diverse array of antigens. The light chains have 1 constant region, which I'm circling right here, and then the heavy chains have 3 constant regions, which I'm circling here, all shown in brown and yellow in this diagram. The variable regions end in nitrogen groups and the constant regions end in carboxy groups, and then there are several disulfide bridges that connect these structures together. There's a disulfide bridge right here that connects the heavy and light chains, and another one right here, and then there's a disulfide bridge in the middle that connects the 2 heavy chains. And then finally, it's very high yield to know that this second constant region of the heavy chains, which I've noted CH2 here, is where complement binds when antibodies fix complement. In addition, you should know that this interface between the second and third constant region on the heavy chains is where macrophages can bind antibodies via their FC receptors. It's also where the bacterial virulence factor protein A binds antibodies in order to protect the bacteria from immune system recognition. B cells are very diverse. It's fascinating to think about the fact that even though we may never be exposed to smallpox or leprosy, all of us have in our body right now B cells that are capable of reacting to those pathogens. We have millions of B cells with unique antigen receptors, including to antigens that we have never seen. And one thing that puzzled scientists for a long time is that there are more unique B cell receptors than there are genes in the entire human genome. At the time, the thinking went that one gene coded for one receptor. So how is this possible? How can we have more B cell receptors than we have genes? Well, this problem was solved several decades ago by realizing that our body rearranges genetic building blocks to make the diversity of B cells. And this is very similar to the way that we use 20 amino acids to create millions of proteins. We assemble them in different mix and match ways to get lots and lots of proteins. The same is true for B cell receptors. We take a small number of genes and we arrange them in lots of different ways to get millions of different B cells. So let's go back to our picture of the B cell receptor. This purple portion of the heavy chain is the variable portion. It's the portion that varies from one heavy chain to another, and it's encoded for by 3 genes. One's called a V gene, one is called a D gene, and one is called a J gene. And then the constant portion of the heavy chain, which is the yellow and brown part here, is encoded by a gene called a C gene. If we zoom in and look at the DNA of a developing B cell, we would see something like what's shown on the screen here. Among the genes that can potentially be used to synthesize a heavy chain, we would see a number of potential V genes. In this example, I've only shown 3, but as we'll talk about in a minute, actual B cells have more than 3 choices for the V gene. We would also see a couple of choices for potential D genes and a couple of choices for potential J genes. And then in addition to this, there are a number of genes that code for the constant portion of the B cell receptor. I'll talk about those later. As the B cell develops, what it's going to do is first join together one of the D genes and one of the J genes. It will then join together one of the V genes with the D and the J, and it will splice out the genes that it does not use. As a result, when mRNA is ultimately synthesized for the B cell receptor, among many potential choices of V, D, and J genes, only one of each will be used. And this is the way that B cells generate lots of different types of receptors. In other words, this is the way there's lots of diversity, because we start with many different choices, and each B cell will make different combinations of those choices, and this will generate lots of diversity in the ultimate B cell receptors. So the heavy chain is made by B cells choosing one V segment from 50 choices of genes, one D segment from about 25 choices, and one J segment from about 6 choices, and all of these are found in chromosome 14. The light chain undergoes a similar process. It has V and J choices that it chooses from, and then random combinations of heavy and light chains add more diversity beyond just the process of which ones of these genes the B cell has chosen. The key point here is that you start with a very small number of genes, but you end up with millions of diverse receptors. And this is the way our body creates millions of unique B cell receptors from a small number of genes. There are two types of activation for B cells. The first way that B cells can be activated is called the T cell dependent pathway. And this only works for proteins because, as you know, only proteins can activate T cells. The other type of activation is called T cell independent, and this occurs with some non-proteins, and this does not involve T cells as the name implies. For the T cell dependent pathway to work, two signals are required. First, the B cell receptors must be cross-linked by an antigen. We'll talk about that in a minute. And secondly, T cells must bind to stimulate the B cell. So pathogens that can activate B cells typically have repeating units of some type of molecular structure, as I've shown right here. And the B cells have a number of receptors scattered around the cellular surface. What the pathogen will do is bind up multiple receptors and cluster them together. This is a process called cross-linking. And the more B cell receptors that get bound and clustered together, the stronger the signal will be to activate the B cell. For T cell dependent activation of B cells, a second signal is required to activate the B cells in addition to binding of the B cell to its antigen, and that second signal comes from T cells. The process begins by B cells presenting antigen to T cells via MHC class II, and the MHC class II can bind to the T cell receptor to activate the T cells. In addition to this interaction between B and T cells, other T and B cell interactions also occur that are very important for T cell dependent activation of B cells. One of those is binding of CD40 on B cells to the CD40 ligand on the T cell. This binding is required for class switching so that B cells can switch from producing IgM antibodies to make other types of antibodies. We'll talk more about how that process occurs in a minute. Another important interaction for T cell dependent activation is the binding of B7 on the B cells to CD28 on the T cells. This binding is required for stimulation of T cell cytokine production, and it is those cytokines from the T cells that drive the T cell dependent activation of B cells. So that's a lot to remember, so I've drawn a picture here to help remind you of all the interactions that are necessary for T cell dependent activation of a B cell. First, the B cell receptors must be cross-linked by the appropriate antigen. Next, MHC class II must bind the T cell receptor via antigen presentation by the B cell and CD4. Next, CD4 ligand must bind to CD40, and this is very important for class switching of the B cell antibody production. And then finally, B7 must bind to CD28, and this interaction is important so that the T cell can begin to secrete cytokines. Sometimes you can have a variation on this process where a macrophage presents antigen to the T cell and activates it via MHC class II and CD4, and then separately the B cell binds to the T cell via CD40, and in this way you can also activate the T cells to secrete cytokines, but the B cell doesn't have to be the antigen presenter. The most powerful and potent way to activate B cells is through the T cell dependent pathway with T cell help. But there is another way that B cells can be activated, and it doesn't involve T cells. So it's called, obviously, the T cell independent method of activation. And there are some pathogens that have so many repeating molecules in their structure that they can cluster a large number of B cell receptors together and activate the B cell without T cell help. And this is called T cell independent activation. A couple of very important points about this process. The first important point is that this is very important for non-protein antigens, especially polysaccharide capsules of bacteria and lipopolysaccharides. If the only way to activate B cells was with T cell help, then B cells could only respond to protein antigens because T cells only respond to protein antigens. But because of this alternate T cell independent pathway, B cells can sometimes react to non-protein antigens. A second key point about the T cell independent response: it's a relatively weaker response, especially compared to the T cell pathway. It's mostly IgM, and there is no memory from this B cell activation process. So on this slide, I've summarized the differences between the two methods of B cell activation. This is the T cell dependent pathway over here. It's for protein antigens only. It generates a vigorous response. The B cells will easily class switch and make IgA and IgG and IgE, and there is memory produced. In the T cell independent pathway, B cells can react to non-protein antigens. It's a relatively weak response of mostly IgM, and there's no memory. And as I said before, this is very important for B cell response to polysaccharide capsules and lipopolysaccharides. There's a very important clinical correlation of the two modes of B cell activation, and it has to do with conjugated vaccines. If you vaccinated someone with a polysaccharide antigen, you would get a relatively weak immune response. That's because you would not activate T cells, because only proteins can activate T cells, and you would have to activate the B cells through that T cell independent pathway, and that pathway produces poor B cell memory, and you get a relatively weak response and therefore weak protection against the antigen. However, if you conjugated the polysaccharide antigen to a peptide antigen, you could then get a much more vigorous response. The B cells would generate antibodies to the polysaccharide, the protein antigen would get presented to T cells, and the T cells would be activated and they would boost the B cell response. Thus you would get a much stronger immune response and much stronger protection. And this is the principle behind conjugated vaccines. We have vaccines against H. influenza type B and Neisseria meningitidis and strep pneumonia that are conjugated. They are pieces of the polysaccharide capsules of these three bacteria conjugated to a protein. Usually it's a toxoid of a bacterial toxin. And the conjugated vaccine produces a much more vigorous response than the polysaccharide capsule components would alone. And that's because you are using T cell dependent activation of the B cells rather than the weaker T cell independent pathway. Now let's talk about the surface proteins you find on B cells. It's important that you know these, especially for step 1s. The first group are proteins that are important for binding with T cells, and we've already talked about all these. You can find CD40, which binds with CD40 ligand on the T cell. You can find MHC class II, which binds with CD4. You can also find B7, which binds with CD28 on the T cells. Some other important surface markers are CD19 and CD20 and CD21, and these are all close to the number 20. That's how I remember them. You can find CD19 on all B cells. You can find CD20 on most B cells, although not on plasma cells. And then finally, CD21 is a receptor for the complement pathway. B cells can bind to complement proteins to bind more effectively to antigens. It's also very important because CD21 is the receptor that the Epstein-Barr virus uses to gain entry into B cells. Now let's talk about the antibodies. So the goal of the activated B cell is to convert into what we call a plasma cell, which is a B cell that secretes antibodies into the plasma and the tissues. And there are several different types of antibodies that B cells can make. The first type are the monomers that look like this shape right here. So IgD, IgE, and IgG are all monomers. The other type that the B cell can make is a dimer. This is what IgA looks like. And then the final type is a pentamer, which is IgM. And all of them have copies, one or more copies, of this B cell receptor right here. And you should remember that the antibody class is determined by the Fc portion of the antibody. This is the Fc portion right here. If it's by itself, it's a monomer. If it's connected to another one, it's a dimer and it's IgA. And then if 5 of them are strung together, it's a pentamer and it's IgM. But it's always this Fc portion that determines what class of antibody you're dealing with. There are 3 functions that antibodies perform that are helpful in eliminating pathogens from the body. The first function is opsonization. This is attracting other white blood cells which will kill the pathogen. So antibodies mark pathogens for phagocytosis. The second function is neutralization. Antibodies can block adherence to structures of pathogens. So for example, if those large IgM pentamers coat a virus, then the virus can't adhere to structures and get inside of cells. And then the third function is that they can activate complement. The classical pathway of complement activation is via antibodies, and I talk about this in more detail in the module on complement. Some bacteria have an important virulence factor that protects them from antibodies, and this is called protein A. It's especially important for Staph aureus. You can just remember the A is for Staph aureus. Protein A is part of the peptidoglycan cell wall of Staph, and it binds the Fc portion of IgG antibodies. So if you have a bacteria here that is Staph aureus, what IgG antibodies will normally do is come in and bind to the surface, and they will adhere like this. And this portion out here, which is called the Fc portion, can attract macrophages and other phagocytes. What Staph aureus will do via protein A is use protein A to bind the Fc portion. It will turn the antibody upside down. Now the Fc portion right here, which normally would attract phagocytes, is hidden from the phagocytes that may be floating around near the Staph aureus, and thus it's protected from the antibody. The antibody cannot opsonize Staph aureus for phagocytosis by macrophages and other cells. So I've summarized this for you here. It prevents macrophage opsonization for phagocytosis. In addition, that Fc portion of the antibody IgG is important for complement activation, and complement activation also won't occur when protein A binds to the antibody. Class switching is the process by which activated B cells change the type of antibody that they produce. And it's very important that you know that activated B cells initially produce IgM only. They also make a small amount of IgD, but the significance of IgD is not clear, so we don't really focus on it very much. However, as the B cell matures and proliferates, it can switch classes. Gene rearrangements will produce IgG, IgA, or IgE instead of the initially produced IgM. You should note that this results in no change in antibody specificity. When you change class, you only change the Fc portion of the antibody. The antigen binding portion remains the same. And the triggers that tell a B cell to class switch are the locally produced cytokines. So for example, IL-4 and IL-5 from the Th2 response can trigger the B cell to switch classes. In addition, when the T cell binds to the B cell in T cell dependent activation, the CD40-CD40 ligand interaction triggers class switching as well. So let's go back to our slide on VDJ rearrangement. This is the slide where I discussed how B cells synthesize the heavy chain by choosing from one of several possibilities for the V gene, the D gene, and the J gene. Well, right after these variable regions on the chromosome come the constant regions, and they are denoted C sub M, C sub D, C gamma, C alpha, and C epsilon. Well, the first two regions, CM and CD, code for IgM and IgD, and that's why IgM is always synthesized first, because the constant portion for IgM comes right after the variable regions. When B cells class switch, what they will do is splice out the CM and CD genes and begin transcribing either the gamma gene, which makes IgG, or the alpha gene, which makes IgA, or the epsilon gene, which makes IgE, and then it will begin synthesizing a different type of antibody. But because of the location of the constant portion for IgM, IgM always comes first. Now let's talk about the individual classes of antibodies, and we'll begin by talking about IgM. IgM is a pentamer, as shown on the screen here in this drawing. It is a collection of 5 B cell receptors linked together. As we just discussed, it's the first antibody that is secreted during an infection. IgM is famous as being an excellent activator of the complement system via the classical pathway. I talk about the complement system in another video, but by way of review, for the classical pathway to activate, 2 complement proteins C1 must link together. Well, C1 binds to the Fc portion of B cell receptors, and you can see that there are lots of Fc portions very close to one another within a molecule of IgM. This makes it very easy for 2 C1 proteins to get next to each other and for the classical pathway to activate. IgM has 10 binding sites. This is the most of any antibody. This is a binding site here. This is another one here. There are 10 of them in each molecule of IgM. For this reason, IgM is said to have the greatest avidity of all antibody classes. Avidity is the total amount of antigen that an antibody class can bind, and IgM can bind the most. Now, just to clear up a point of confusion here, a separate term from avidity is the term affinity. Affinity is the strength of an individual binding between an antibody receptor site and an antigen. So the strength of this binding to one site is called the affinity of an antibody for an antigen. Many other classes than IgM often have greater affinity for binding antigen, but they do not have greater avidity because they don't have as many binding sites. IgM often prevents the attachment of pathogens. That's how it works. It's a big molecule, so you can imagine that if it attaches to a bacteria, for example, it will get in the way of that pathogen's ability to attach to surfaces within our body. IgM is a relatively weak opsonin. That's because many other elements of the immune system have Fc receptors which bind to the Fc portion shown here on the antibody. Well, all the Fc portions in IgM are clustered together in the middle and not exposed. Therefore, they often do not bind Fc receptors in other immune system elements, and therefore IgM is a relatively weak opsonin. It can, however, opsonize to some degree because it can activate complement, as we just discussed, and the complement pathway can generate C3b, which is an opsonin, so there is some opsonizing ability of IgM. And then finally, because it's a large pentamer, it cannot cross the placenta. This means that IgM cannot confer antibody benefits to the fetus from the mother. IgG is a monomer. This is a picture of IgG right here. It's not a pentamer like IgM. It has two antigen binding sites. It's called a divalent antibody for that reason. There are 4 subclasses of IgG called IgG 1 through 4. And this is the most important antibody in a secondary response. When you've been exposed to a bacteria or a virus and you develop memory B cells and then you are re-exposed, the secondary response is rich in IgG antibodies as opposed to the primary response. This is the only antibody that crosses the placenta, and therefore it's the most abundant antibody in newborns. Its greatest feature is that it's an excellent opsonin, especially IgG1 and IgG3. Macrophages can bind to this Fc portion right here very well, and this helps them to identify and consume bacteria. It's the longest lived and most durable of all the antibody types. It can survive for several weeks in the body, and it's the most abundant class you will find in the plasma. Most of the antibodies in your plasma are IgG. IgG is very important for encapsulated bacteria. As you may know, the capsule of some bacteria allows them to resist phagocytosis by macrophages and neutrophils. However, once they're coated with IgG, the IgG molecule serves as an opsonin to draw in the macrophages and neutrophils, and this leads to phagocytosis. And this is a very important way by which you clear encapsulated bacteria from your body. IgA is the main antibody of mucosal surfaces and mucosal secretions, so you will find it in the GI tract and the respiratory tract. You can also find it in saliva and tears. It's a monomer in the plasma, but then it crosses epithelial cells by a process called transcytosis and gets transported through the cells. And as it goes through those cells, it gets linked by something called the secretory component, which is represented by this little link here, and then it becomes the dimer that you find in the secretions. Because it's linked by the secretory component, it does not fix complement. However, it's excellent at coating mucosal pathogens. This makes it the ideal antibody for mucosal surfaces. It will coat the pathogens so they cannot invade. The pathogens will get swept away with mucosal secretions, and because it doesn't fix complement, there's no inflammation and therefore no destruction of the mucosal layer. It's for this reason that it's secreted into mother's milk so that the baby will consume this and get lots of IgA in its GI tract to protect it from pathogens as a newborn. This is another picture of an IgA molecule. It has a joining segment and then it has this long snake-like secretory component which is synthesized by epithelial cells, and it needs the secretory component attached to it in order to be secreted across the mucosa. There are some bacteria that have a virulence factor called IgA protease. This is an enzyme that cleaves IgA secretory component and makes the antibody less effective. It allows the bacteria to colonize mucosal surfaces, and this is an important virulence factor for strep pneumonia and H. influenza and also both types of Neisseria. The last antibody we'll talk about is IgE. The purpose of IgE is to bind to mast cells and eosinophils. It's designed for defense against parasites. If you think about a very large helminth or worm, this is way too big for phagocytosis by a macrophage. So IgE will bind, and as a result of the binding, mast cells or eosinophils will degranulate their contents onto the parasite to try and kill it. There's a low concentration of IgE in the plasma, and it does not activate complement. And its major medical importance, especially in Western societies, is in allergic reactions. Most people aren't exposed to many helminths or parasites, but many people have allergic reactions, and IgE is important for mediating seasonal allergies and anaphylactic shock. And I talk about this in more detail in the module on hypersensitivity reactions. Here's an illustration of 2 common ways that IgE is involved in immune responses. The first has to do with mast cells. So mast cells will bind IgE antibodies on their surface. They will bind the Fc portion, and the Fab portion will be sticking out. And then a parasite, or more commonly an allergen, will cross-link these antibodies together, and then the mast cell will degranulate into the tissues. In the case of eosinophils, Ig antibodies will bind to a helminth. This purple line is meant to represent a helminth. And then the eosinophil will bind to the Ig antibodies, and then it will release its contents to try and kill the helminth. And this is an example of the process of antibody-dependent cellular cytotoxicity. Let's talk about a very important mechanism by which B cells hone their effectiveness in eliminating pathogens, and this is called somatic hypermutation. This is a late event. It's a process that B cells undergo during inflammation and infection, often after they have already class switched. There are certain portions of the V, D, and J genes that I described before which have a very high mutation rate. And B cells must be re-stimulated for ongoing proliferation. So what is going to happen is, as the B cell proliferates during an infection, mutations will occur in the V, D, and J gene. Only the strongest binding B cell receptors will lead to the most proliferation. The result is that over time you get stronger and stronger binding of the B cell receptors and the antibodies. This is a process called affinity maturation. The receptors mutate and you get stronger antigen binding over time. If that's confusing to you, here's a picture to help you understand. So imagine that you have a B cell and it is proliferating during an infection. It will have small mutations occur in those V, D, and J genes. Some of those mutations will result in a new receptor that is relatively weak binding. Others will also result in a mutation that is relatively weak binding. But some of those mutations will result in a slightly different receptor that has stronger binding. The stronger the binding, the more proliferation you get. So this B cell in the middle with the strongest binding will proliferate the most, and then the process will repeat itself. So as a result of somatic hypermutation, as the B cell proliferates during infection, you get stronger and stronger binding of the B cell receptor and the antibodies. Let's talk about the fate of B cells once they are activated. So after activation, B cells have a choice. They can either become a plasma cell, which makes antibodies, or they can become a memory B cell. Plasma cells usually travel to the spleen or bone marrow, and they can secrete thousands of antibodies per second. Their metabolic activity is remarkable. They die after a few days. However, more will be created if the infection or the antigen persists and continues to drive production of plasma cells. The other option for what an activated B cell can become is a memory B cell. And remember what I said before, only T cell dependent activation produces memory. The T cell independent activation of B cells does not produce memory. So we've talked about a lot of things that B cells can do as they develop, and here I've shown you a timeline just to help you keep straight when all these processes occur. So in the bone marrow, before you're ever infected or exposed to an antigen, that's when B cells develop and do the VDJ rearrangement to choose the B cell receptor that they will have on their surface. In the lymph nodes during an infection, that's when you get class switching. That's also when you get somatic hypermutation. And then after the infection has gone away, this is when you're left with just memory cells present in the body. The reason memory B cells are so helpful is because on a second exposure to the same antigen, you get a much quicker and more vigorous antibody response. So on this slide, I've shown a primary antigen exposure down here, and it takes several days to begin to generate an IgM and an IgE response. When you get a second exposure, you get a much quicker and much more vigorous response that is mostly composed of IgG antibodies. And remember, those IgG antibodies are those excellent opsonizers, and they will go and more effectively eliminate the pathogen than in the primary response. So up here I've summarized for you, the primary response is slower and weaker, the secondary response is faster and stronger. I'll finish this module by talking about vaccines. So vaccines are designed to elicit a B cell and T cell response without an infection, and they provide protection via immune memory. And there are several different types that you need to know about. There are live attenuated pathogens, killed pathogens, and then oral and intramuscular pathogens. Live attenuated vaccines contain pathogens that are modified to be less virulent, and they tend to induce a strong cell-mediated response, especially they generate a T cell response, and this response tends to be more durable and long-lasting. The downside is that live attenuated vaccines carry some risk of infection, especially if you administer them to immunocompromised patients. In addition, you often have to give them after children are a year old, because younger than a year old there can be maternal antibodies present that can kill the pathogen. And the classic example of a live attenuated vaccine is the MMR vaccine. MMR stands for measles, mumps, and rubella, and it's a combination of 3 attenuated pathogens that are responsible for the immune response. Killed vaccines have a pathogen that's been killed, but the antigens remain intact, and these tend to elicit a strong humoral response, especially in terms of antibodies. Unfortunately, it's a weaker immune response than the live attenuated vaccines, but the upside is there is no risk of infection because the pathogen has been killed. If you give a vaccine orally, you will stimulate the GI mucosal immune system, and this will generate a largely IgA response. And this is useful for pathogens that get into the body through the GI tract. So for example, there are oral polio and rotavirus vaccines, and this makes sense because you stimulate lots of IgA antibodies in the GI tract, and as a result, these bugs can never get into the body. If you give a vaccine intramuscularly, you will stimulate a tissue response. This tends to be largely IgG. All the types of vaccination I've been talking about up to now are examples of active immunization. And it's called active because you require the patient's body to actively generate an immune response. There is another type of less common immunization called passive immunization. This is where you directly administer antibodies to the patient. This will provide only short-term protection for a few weeks. There's no memory or long-term protection, and it's only used for dangerous imminent infections. So for example, if you think a patient may have rabies or tetanus, these are very dangerous diseases, so you will administer antibodies against either the tetanus toxin or the rabies virus to try and neutralize things before the patient gets sick. Passive immunization is also the way that the mother helps out the fetus. There are maternal antibodies passed on to the fetus, and this is an example of passive immunization of the baby. And sometimes passive and active immunization are done simultaneously. So for example, if you think a patient might have been bit by a rabid animal, you will administer rabies immune globulin to try and neutralize any virus that might be inside the body. You'll also give the person rabies vaccine to generate memory and long-lasting infection. And that concludes our module on B cells.